Hello everybody, bonjour à tous. To introduce this new session, I want to start with an observation. Today we live in a world where we collect more and more diverse data. And this is the case in medicine, where we acquire a huge amount of data over a very long period of time. And did you know that 90% of the medical information collected is no longer used after a year? In fact, a great deal of information is sometimes little or poorly used. But there is another problem. We are living long. In France, for instance, the life expectancy for women is 85.3 years, and for men is 79.3 years. But the life expectancy in a good health is still too low. In France, again, for women, life expectancy in a good health is 64.1 years, and for men is 62.7 years. So we have a problem because this means that we live on average almost 20 years with a health problem. And the life expectancy in a good health in the north country of Europe is greater than 10 years in comparison with country in the south of Europe. And we can explain that thanks to the prevention. And with the Meditwin project, we are using modeling and simulation to increase the number of life in a good health for diseases that currently account for 33% of DALYs worldwide. So Meditwin is a 5-year project based on a consortium with 14 partners, with 7 instituts hospitalo-universitaires, 1 university hospital, 4 startups, INRIA, and Dassault Systèmes as lead partners. And the aim of this project is to develop components of the human virtual twin in the form of software as medical device, covering different therapeutic areas like neurology, oncology, and cardiology, all based on a collaborative platform that facilitates the sharing of information between healthcare professionals. And before I give you a concrete example of medical use in Alzheimer disease and vascular dementia, I'm going to introduce Guillaume Turpin from INRIA. And Guillaume, Meditwin is a partner, INRIA is a partner of Meditwin. How is the project Meditwin important for your structure?
Thank you, Baptiste. Well, as you know, INRIA is the French research center in applied mathematics and computer science. We are about 4,000 people in 200 teams, and we aim to perform research at the world-class level and social and economical impact of that research, which is a key point of our activities. And Meditwin is perfectly aligned with those goals. We have 10 research teams involved in Meditwin, and the project is designed so that we go from research to market with the software as medical device you mentioned just a few minutes ago. So this is a project perfectly aligned with our strategy. And second point is that this project is big, ambitious, and for us with a special partner, which is Dassault Systèmes, that I'm very proud to thank today, because as I said earlier, INRIA is the research center of applied math. And Dassault Systèmes is the kind of biggest French company that design products and services thanks to applied math. So Dassault Systèmes is one of our strategic partners, and maybe the biggest one. And Meditwin is the biggest project of that kind with industrial partners that INRIA has even done. So for all those reasons, of course, Meditwin is a very important project for INRIA.
Okay, thank you, Guillaume. I have another question. How will the Meditwin project be a transformative project for your research ecosystem?
As Meditwin is designed to go from research to market, and because of its size, we have two challenges in Meditwin. Three. Top class, world-class research level, of course. And then we have to be able to deliver what is expected from us. And above research, we have to deliver reusable and transferable softwares. So we have to, this is a challenge because as we have 200 different teams, those teams, they used to work on their way. But we have to rethink our engineering processes so that we can collect something from all those softwares and make them more industrialized and go a step further in our way to design transferable softwares. And this is from the engineering part. And on the organizational part, we have different entities in INRIA: research direction, transfer and innovation directions, the newly created program agency, and we have 9 research centers all around France and the headquarters, and all those people have to work together. We have a core team of 5 people, but indeed, more than 50 people work for Meditwin in INRIA in the support teams, not in the above the research teams. And we have to hire more than 70 people during the project, PhD students, postdoctoral students, and engineers. So on the organizational side, we have to improve our processes also.
Okay, thank you, Guillaume.
Okay.
So now is the turn of my colleague Louise Moreau.
Thank you, Baptiste, for introducing our open discussion on Alzheimer's disease. So for this session, I am joined by Dr. Nicolas Villain, who is a neurologist at Hôpital Pitié-Salpêtrière and also a researcher at the Paris Brain Institute, and Dr. Ninon Burgos, who is a researcher in the Paris Brain Institute in the Aramis lab. So just as an introduction, Alzheimer's disease is a slowly progressing neurodegenerative disease, which is the main cause of dementia in the world and which affects currently around 50 million people worldwide. And this number is estimated to be multiplied by 3 by 2050. So while the first treatments begin to arrive in the United States after decades of failing attempts, the early diagnosis of the disease remains a challenge in a society whose population is aging more and more. And so, Dr. Villain, you're a neurologist. Could you tell us a little bit more about the medical challenges surrounding Alzheimer's disease?
Thank you, Louise, for the introduction. Good morning, everyone. Thank you for highlighting Alzheimer's as a case study in this Meditwin project. We are very enthusiastic that Alzheimer's could also be highlighted today. So current medical challenges, they are huge. I mean, as you mentioned, the figures are huge. Worldwide, we're talking about 50 million individuals, and we have no cure at the moment. So we have everything to do. And I will focus today in our roundtable on the challenges we'll try to tackle within this Meditwin consortium. Of course, we will not solve the Alzheimer's disease problem worldwide. I wish we could, but let's be realistic. So we'll focus, however, on a very crucial question, which is the diagnosis and the early mechanisms of the disease. So just a few slides to illustrate what I will discuss today. So what is Alzheimer's? Alzheimer's disease is a neurodegenerative disease, as you mentioned. So you have neuronal loss. You also have brain atrophy. Sorry, the illustration is in French, but you will translate. So you have synaptic loss, neuronal loss, and you have two protein aggregates which are abnormal, which are made of tau protein, and we call them in French dégénérescence neurofibrillaire, neurofibrillary tangles in English, and amyloid plaques, beta amyloid peptide is being accumulated between neurons. So these are the main features, and now we can dose them, these abnormal protein aggregates, for diagnosis within the CSF, the cerebral spinal fluid. And we have an increase in the tau protein concentration and instead a decrease in the beta amyloid peptide concentration. So that's the state of the art for diagnosing Alzheimer's disease today. So you have to perform cognitive testing, you have to dose these biomarkers, and then you have a precision diagnosis. And so we have moved from this diagnosis of individuals who have symptoms, come to your consultation, tell you, okay, doctor, I experiencing memory loss. Is it Alzheimer's? So we do cognitive testing and we do this biomarker in CSF dosing, and we've realized that this biomarker changes actually occur years and even decades before the first symptoms happen. So let's illustrate this with Chinese findings that were published in the New England Journal of Medicine this year. They have performed longitudinal testing of CSF in volunteers for about 20 years, and they realized that you have a CSF A-beta 42 changes, which are the very earliest changes that happen, you see, about 17 years before that. So, you know, with Alzheimer's now, we not only can diagnose the disease, but we can also anticipate it even before symptoms and neurodegeneration happen. So this looks great. This looks even greater since now we are transitioning to blood. CSF requires lumbar puncture, which is, of course, inconvenient. So let's use blood instead. And can we do that? Yes, we can. We can also dose the same proteins, tau and A-beta, in blood. Of course, the concentration is a bit lower, like 20 to 100 times lower. So that's the challenge. And it's been a challenge for decades. And since the last 5 years, we have a second generation of immunodetection, immunoassays, that have been released by many fabricants, including Quanterix, Roche, MSD, Shimadzu, that allows you this increase in detection level. And this increase in detection level allows you to get to this 100 times lower concentration of tau and A-beta in the blood. So now we can dose this protein in the blood, and we have very recent results that show us on the left-hand side of this slide that we have very good diagnostic performance compared to amyloid PET and tau PET or CSF, which are our current gold standard for this protein measurement in clinical practice. See this AUC, this area under curve that tells you roughly the diagnostic accuracy. So with these blood biomarkers, here you have several in a list in plasma. We see diagnostic accuracy more than 90%. So it works well, and it works well enough to be used in clinical practice. And using that, we are now transitioning to use them in clinical practice, and we could save 80 to 90% of lumbar puncture or PET scans that are, of course, invasive and costly. So everything looks great on the diagnostic approach in Alzheimer's disease. We are moving to precision medicine by dosing these proteins, and we have more and more easy to implement technique in clinical practice. However, we have some significant drawbacks moving to this biological concept of Alzheimer's, has also a lot of unexpected implications. For example, we realize that actually 75% of individuals who have these abnormal biomarkers are actually cognitively unimpaired, meaning that only a quarter of them have Alzheimer as we think it is today, this neurodegenerative disease slowly progressing to dementia, etc., etc. And this is a problem since less than 50% of these cognitively unimpaired individuals will develop symptoms in their lifetime. So transitioning to blood also means let's be careful about what we are diagnosing and what we are telling to these individuals. And that's what I just mentioned. We have a new population that comes into clinical practice since the vast majority of the individuals with abnormal Alzheimer biomarker will be on the blue line, meaning that they will have these abnormal proteins aggregates in the brain, but they will remain cognitively normal in their entire lifetime and die of something else. Of course, a few portion of them will turn to Alzheimer's, meaning developing neurodegeneration and developing symptoms. So we have this new population that come to light into everyday clinical practice. These are these individuals who will have a blood test for Alzheimer's and will come to see you. Hello, doctor, I have a blood test that is positive for Alzheimer. Do I have to sell my house? Do I have to move to a nursing home? What should I do? So this is actually a problem because we don't have a lot of answer to offer them. They tell you, okay, but what is my actual personal risk of developing symptoms? And the data is very poor because we have had these biomarkers in blood for less than 5 years, so we have very poor data. And this, of course, prospective study that highlight the risk of these individuals are very sparse. And they also include very few individuals. So we have preliminary findings that tell you, okay, you may have these demographics, these genetic variants, it's a higher risk, but we can't quantify it, we can't be sure enough. So that's the question we'll tackle in Meditwin. So we'll start a new cohort to help identifying the lifetime risk factors in this of cognitive decline in these individuals who are cognitively unimpaired but have these Alzheimer's biomarkers. Thank you for listening to this introduction to our goal in this Alzheimer's package of Meditwin.
Thank you, Dr. Villain, for highlighting the medical challenges of the disease. And also, we do understand that there's hope through blood testing, but it does open a new set of scientific questions that we have to answer, and we'll try to do this in the Meditwin project. So now, Dr. Burgos, could you tell us a little bit more about the solution that you've been developing in the Aramis team in order to solve the medical challenges that Dr. Villain has discussed?
Hello, everyone. So I'm Ninon Burgos. I'm a CNRS researcher in an INRIA team, so-called Aramis. And so we've seen that blood is becoming very important. So in Alzheimer's disease, but right now I will present what we do still with imaging that is still useful, hopefully. And I will show you how we can automatically detect lesions within brain of potentially Alzheimer's disease patients. So I will show you many images of that type. So these are PET images overlaid on anatomical MRI images. So here you have the FDG PET of a healthy control. So what shows FDG is a glucose consumption of the neurons, so how much sugar the neurons consume to work. So you here see nicely that all the neurons in the cortical ribbon consume glucose, because you see that the signal here is red for most of the parts. when you have Alzheimer's, you will have what we call hypometabolism. So your neurons will consume less glucose, and this will lead to their death and so to atrophy. What we want to do here is to detect this hypometabolism as early as possible, and so when it is still quite subtle. So here on these patients, you can see it in these regions, for example. So for people used to look at these images, this is like obvious, but the aim is to detect the lesion when they are still quite subtle. So our aim is to obtain maps such as the one on the right, what I call a patient-specific abnormality map that highlights the areas that appear abnormal in the brain. The way we do that is by recreating what we call the pseudo-healthy twin, which corresponds to, in this case, the PET image of the patients if the patients were actually not a patient but a control. But that corresponds to the patient's anatomy. So it's not any PET image that I'm displaying here, it's the PET image of the patient if it were normal. And so here is what we could get as results. So for an AD patient, so we would highlight the areas of abnormality in the brain, here in dark blue on the left. So to do that, you need basically two ingredients. You need to know the anatomy of the patient, here what I call target subject, because you want to recreate the pseudo-healthy twin that is specific to this anatomy, so you need to know the anatomy. And you also need to know what a healthy looking image look like in general, so you need many examples of pairs of anatomy and PET image of healthy controls. So I've been working on this topic for quite a few years, and at the time I was using, like, let's say, old school image processing techniques to obtain these types of pseudo-healthy twins. So what I was doing was registering each control to my target subjects to align all the images, and I was then trying to see which controls were the most similar in terms of anatomy to the patient I was studying, and this at a really local scale, to then combine the PET images of the control subjects, again at the local scale, to recreate my what I call subject-specific models of healthy appearance. Once I have that, what we can do is compare this model to the real PET image of the patient that we have. And here, because we have the real image of the patient, an average, basically a weighted average and a weighted standard deviation, what we can do is compute a z-score. And this is what you have on the right. It's for each voxel, you have basically a z-score that tells you how much each voxel differentiates from the normal. So if we look at some results, so first for a cognitively normal person, so here I first display the PET image overlaid on the MRI and the abnormality map. So here again, each voxel corresponds to a z-score. You have the scale bottom right. And so here you don't pick up anything abnormal, which is expected as this is a cognitively normal subject. When you progress into the disease, first early mild cognitive impairment, late mild cognitive impairment, you start seeing lesions appearing in the brain until the Alzheimer's disease stage. Here I've shown you example with FDG PET, so that shows you glucose metabolism, but the same principle can be applied to other PET tracers, such as the amyloid one, that shows you the accumulation of amyloid plaques in the brain. So you have the four same subjects, and you see the amyloid spread in red, so as you progress into the disease. So this is what we've been doing until now. But of course, these days you don't do like old school like image processing. You use like AI to do all that. And so in the Meditwin project, we will continue our work on using deep generative models to generate these pseudo-healthy twins. So we will develop either like GANs, which we already do, or more like recent deep generative models to generate pseudo-healthy twins from the anatomy. So during the training phase, basically what happens is that you train a network to generate pseudo-healthy PET images only for healthy controls. So you teach like the network to only reconstruct pseudo-healthy images. So that's at the application phase. When you provide an anatomy, then the network only reconstructs something that appears healthy because it's the only thing it knows how to do. And again, you can compare to the real PET image to generate the abnormality maps. Here I've shown you many examples with FDG PET, but of course we'll move on also to tau PET, which is more like up to date in the study of Alzheimer's disease. Thank you.
Thank you, Dr. Burgos, for presenting this solution of quantitative analysis of PET. And as you mentioned, the goal in the Meditwin project will be to improve and adapt this solution to new PET radio tracers specific for Alzheimer. So at Dassault Systèmes, we're also providing an answer to those medical challenges by developing the living brain twin. So we develop a twin which is completely personalized, built from multimodal patient information, and which allows computing a large number of biomarkers, all of which can be visualized within the living brain viewers that we're developing in the twin. So if we could have the video launched, please. So, for example, the biomarkers that we could include are the morphometry measures of cortical and subcortical areas of the brain, the standardized uptake value ratio computed from PET scan, but also the memory network, as you can see on your screen. All of those biomarkers are extracted from combined imaging modalities, and we could also integrate biomarkers computed by our Meditwin partners. So, for example, the maps that you're computing for PET, or for example, the EEG analysis that other partners are computing. So the living brain viewer allows having an integrated visualization of all those biomarkers which are personalized for unique patients. And this enables having a better analysis and a better collaboration of the medical staff, which is involved in the patient care network, in order to provide a better informed medical decision. So given the solutions that have been presented, Dr. Villain, are you hopeful for your future in the medical practice and in Alzheimer research?
Absolutely. What you just mentioned and what Ninon mentioned will allow you to collect this large-scale, multi-level scale that we've been discussing this whole morning in these individuals who are normals, and some of them are on the path of Alzheimer's, some are not. So collecting all this data at multi-scale level will allow us to then describe and set an algorithm of prediction. And for each individual, according to this set of data that we'll collect, also involve EEG in this project. We also involve a vessel measurement within a retina. So we'll have really a lot of data set collected for these cognitively unimpaired individuals that will be followed up for 5 years. And then we'll be able to stratify this information and create an algorithm that will allow us to tell these individuals who will come and tell you, hey, doctor, do I have Alzheimer? My blood test is positive. Yes, you will have. No, you will not. What is my percentage of risk in this 5, 10, 15 years? And this will be a huge step for us. It will also be a huge step for treatment development, because if you can then stratify your treatment development according to the risk of your individuals, it's more than important. You were mentioning earlier this late approval of anti-amyloid therapy in the US that was refused by the EU this summer. You know why? Because the risk-benefit ratio is not incredible at the moment. We have bleedings and edema of the brain. So if we consider giving these drugs to asymptomatic individuals who only have 20% risk of developing symptoms in their lifetime, maybe it's a bad idea. So we need this percentage, because instead, if I tell you, okay, now you have 95% risk of developing Alzheimer's, I may consider taking the risk of 5% risk of edema or hemorrhage, whereas I will not if I only have 20%. So we are moving to precision medicine, precision therapy, and Meditwin will help us tackle that.
Thank you, Dr. Villain. Thank you, Dr. Burgos, for your insight on the disease. And also, as part of on this use case on Alzheimer, we are joined by Foresight Institute and also 2 startups, which are Kernel and Neurometers. And we are thrilled to begin this new project. And Baptiste, I leave you the floor to conclude this Meditwin session.
Yes, thank you, Louise. Thank you for this presentation. To conclude, so in addition to the neurological field, Meditwin, the project Meditwin will address other use cases in other therapeutics area like cardiology and oncology. And the aim is to start with very specific use cases like rare disease, neurodegenerative disease in a given therapeutic area, and then extend the development of the component of the virtual twin in this given therapeutic area. The aim of the virtual twin is to integrate the complexity in all its diversity into a multidisciplinary, multi-scale, and longitudinal explanatory scheme using a systems approach. The virtual twin aims to share complex medical information between different players involved to facilitate collaboration between the different medical bodies and the different monitoring sites. So now Guillaume will present you 2 of our dreams with the Meditwin project.
Okay, thank you, Baptiste. Yes, we may say we have 2 dreams. The first one on the research side is to go towards a clinical trial virtual twin, which will be a guide for real clinical trials. And the second one is to make the patients, more at the center of their own health, thanks to the digital twin. And with this digital twin, giving medical practitioners the state-of-the-art know-how in order that the whole society will benefit from this personalized medicine.